Hi. So today we're going to discuss how can mitochondrial defects or mitochondrial dysfunction lead to type 2 diabetes. So you know that diabetes type 2, the person is either not releasing enough insulin or not producing enough insulin, or the person is developing insulin resistance. The body is not able to respond to insulin. So today we're going to focus on how can mitochondrial defects or mitochondrial dysfunctions impair the release of insulin by the pancreatic beta cells. Now, what are the risk factors contributing to the development of diabetes? We have obesity, we have high lipid levels, inactivity, age, and much, much more. High lipid levels, if we have a high LDL levels and low HDL levels, this will increase the risk of developing diabetes. Overweight, not exercising, all of these factors can increase the risk of developing type 2 diabetes. Today we're going to focus on obesity, hyperglycemia, and high lipid levels, how they could influence mitochondrial dysfunctions, and how they could influence type 2 diabetes. So, now let's talk about how insulin is released by the beta pancreatic cells in the normal state, non-pathological state. If we have a normal person, what takes place is we have glucose rushing into the beta pancreatic cells through the GLUT2 channels, glucose 2 channels. Glucose through the glycolysis reactions, it reaches then the mitochondria as pyruvate, and the normal cellular respiration reaction is taking place. Now we reach the electron transport chain. Here we are focusing at the inner mitochondrial membrane. During the electron transport chain, what takes place is that protons will be pumped from the mitochondrial matrix into the intermembrane space, and then they will rush through the ATP synthase enzyme to produce ATP. So this is what's happening in the normal state. After ATP is produced by ATP synthase enzyme, this is the ATP synthase enzyme. ATP is produced by ATP synthase enzyme. ADP, ATP transporters are used to transport ATP out of the mitochondria, and ATP will be used to close ATP sensitive potassium channels. So these ATP sensitive potassium channels will close in the presence of ATP. When the ratio of ATP to ADP increases, these potassium channels will close, and what will take place is that we will have a higher cytosolic potassium ion concentration. When this takes place, when these channels close, the potassium ions will not be able to pass out of the cell and maintain a resting membrane potential. So what takes place is that the membrane potential will become more positive and depolarization will take place. So if we draw the charges or represent the charges, the inside of the cell will become positive and the outside of the cell will become negative. So this is what's basically takes place, depolarization. Now, when depolarization happens, voltage-gated calcium channels will open, as what happens in the synaptic presynaptic neurons. The same happens here, is that the voltage-gated calcium channels will open, and this leads to the influx of calcium ions into the cytosol of the pancreatic beta cells. These calcium ions then will drive the exocytosis of insulin out of the beta pancreatic cells into the bloodstream. So this is the process that's responsible to release insulin from the beta pancreatic cells into the blood. Now, if we consider the pathological state, what I mean by a pathological state is that a person is having a mitochondrial defect. Mitochondrial defects could vary. One of the defects could be within the electron transport chain, is that we are having, for example, an increased concentration of superoxide anions, like these ions, superoxide ions. Also, we could have other defects within the mitochondria, and which are the most important, and we will talk about them now. Now, if the person is having obesity, glycemia, and high lipid levels, these factors can increase the expression of a certain protein known as UCP2. UCP2 is called uncoupling protein 2. Now, what's the meaning of uncoupling protein? An uncoupling protein is able to form or embed itself within the inner mitochondrial membrane, and it will form a hydrophobic channel through which the protons or the hydrogen ions can diffuse from the intermembrane space into the mitochondrial matrix. So this what will take place, and the concentration of hydrogen ions in the mitochondrial matrix will increase, and here the concentration will decrease. Now, what's the effect of this? The effects of this is that the proton motive force will decrease. So, what's basically taking place here is that as hydrogen ions rush from the intermembrane space into the mitochondrial matrix through the uncoupling protein 2, the concentration gradient of hydrogen ions will decrease and the proton motive force will decrease. So you could imagine hydrogen ions rushing into the mitochondrial matrix with a lower force or with a lower potential, and this will correspond to reduced ATP synthesis by ATP synthase enzyme. Why? Because most of the hydrogen ions have diffused through the uncoupling protein, and less hydrogen ions will be available to diffuse through the ATP synthase enzyme, and this is an important point to be considered. Less hydrogen ions will diffuse through the ATP synthase enzyme, so less less ATP. If we are having less ATP, okay, and remember that ATP is important in the process of releasing insulin by the beta pancreatic cell. If we are having less ATP, this means that some of the ATP sensitive potassium channels will open. In the presence of ATP, these channels will close. However, in the absence of ATP or with a reduced ATP level, more of these channels will open. So what will take place is that we can draw, for example, here an open potassium channel, K, for example, ATP, and the potassium ions will rush out to maintain a resting potential or to maintain the normal potential within the beta pancreatic cells. So what takes place is that the cell will be hyperpolarized and not depolarized, and the potential or the threshold will not reach to open the voltage gated potassium channels. So these channels will not open, and there will be no influx of calcium ions into the cell. Thus, there will be no exocytosis of insulin out of the beta pancreatic cells. So in this way, this uncoupling protein 2 has caused reduced calcium influx into the beta pancreatic cells in an indirect way by reducing ATP levels. So less insulin is released, and we can see here that one of the causes of type 2 diabetes is that the pancreas is not able to release enough insulin. Now you can focus on this sentence now, that UCP2 is an oxidative phosphorylation uncoupler or decoupler. So it decouples oxidative phosphorylation. It reduces the production of ATP by oxidative phosphorylation, less ATP, and it will generate heat. By the way, it will generate heat. Most oxidative phosphorylation uncouplers lead to the generation of heat instead of ATP and lead to a less efficient cellular respiration process. Other oxidative phosphorylation uncouplers exist. One of them is known as thermogenin, and you can deduce from its name, thermogenin, that it produces thermal energy or heat. So yeah, this is how can an oxidative phosphorylation uncoupler and superoxide ions also can increase the activity of UCP2. So this is how an increased activity of UCP2 or an oxidative phosphorylation uncoupler can lead to type 2 diabetes. Hope you like this video. In the next video, we're gonna discuss how can mitochondrial defects or mitochondrial dysfunction lead to insulin resistance. See you in the next video, and bye.